Before conducting the quantitative analysis, we need to make sure that the analytical method is robust and reliable. Today, I will navigate you through the processes of LC method validation. In this lecture, you will learn how to optimize the ionization and mass parameters during method development and the basics about LC mass quality assurance and quality control. Many factors may affect the signal intensity of the analyte, from sample pretreatment, chromatography separation, to ionization and mass parameters. In the following slides, you will learn how to get the best performance out of these parameters. If your analytical methods includes a sample pretreatment step, such as a solid phase extraction, the process efficiency will be the important parameter in determining the method sensitivity. By testing the different washing and eluting conditions, we can arrive at the optimal SPE protocol by comparing the acquired peak areas or peak area ratios like standard to internal standard ratio. How can we evaluate the process efficiency? By comparing the peak area of neat standard and pre-spike matrix, we can assess the process efficiency, extraction recovery, matrix effect, and the extraction yield in the same experiment. Process efficiency is the quality of the whole analytical process, which is related to alterations due to interferences during the sample preparation and analysis, such as ionization, ion transmission, etc. Process efficiency can be estimated with the ratio of peak areas from the matrix sample spike prior to the sample preparation and from a neat standard. Residual compounds still present after sample preparation can interfere with the mass ionization process, leading to the well-known signal suppression or enhancement situations. The influence of endogenous compounds on LC mass or matrix effect is evaluated by the ratio of peak area from the matrix sample fortified after the sample preparation and the neat standard. This is related to the concept of matrix factor, defined as a ratio of the analyzed peak response in the presence of matrix ion to the analyzed peak response in the absence of matrix ions. Interference may also affect the sample preparation, leading mostly to a decrease in the extraction yield when compared to standards. The recovery of extraction in the presence of the matrix or extraction recovery should therefore be determined with the ratio of pre-spike over post-spike peak areas. However, this calculation does not allow for differentiating a low extraction recovery due to interfering compounds from the poor extraction yield due to the sample preparation itself. Since generic procedures are often used in multi-analyte determination, a low extraction yield could lead to an recovery misevaluation. Therefore, extraction yield as determined by the peak area ratios of neat extraction standard and neat standard can help to clarify whether the low extraction recovery results from the interfering compounds or the SPE itself. The chromatography condition also plays a role in determining the analytical specificity and sensitivity. How can we assess the efficiency of chromatography separation? Well, first we need to know if the compounds have the appropriate interaction with the column. A compound with a weak column interaction could be easily interfered with the co-eluted matrices and salts, where severe ion suppression may be observed in the LC mass analysis. However, the analytical runtime would be too long if the analyte-column interactions were too strong. We can use the retention factor K to evaluate the interaction between analyte and column. Retention factor refers to the time the analyte interacts with the stationary phase relative to the time analyte spends in the mobile phase, which can be expressed with this equation. The greater the K, the stronger the analyte-column interaction. As mentioned in our previous lecture, scanning too many MRN channels in the same time segment would decrease the instrument's duty cycle for each channel, which in turn reduces the analytical sensitivity. Successful chromatography separation would enable one segment for one analyte, which would therefore increase the duty cycle and sensitivity as few ions are wasted. So how do we know the separation efficiency between two peaks? Usually, the separation efficiency of two peaks can be described by the resolution value Rs expressed as a ratio of the distance between two peaks to their average width. The baseline resolution is achieved when the resolution is 1.5. The greater the resolution value, the better the separation efficiency. In summary, there are three major factors that affect the resolution: retention, selectivity, and efficiency. Retention is a measure of the degree of interaction with the stationary phase, which is quantified with retention factor. Selectivity is a measure of the differences in the characteristics of the components in the mixture, as quantified with resolution value Rs. Efficiency is a measure of the sharpness of the peak, which can be quantified with a signal-to-noise ratio. Let's showcase how to apply these parameters into the method development. Assume that you are developing a LC mass method for two analytes A and B, and you are considering C18 or C8 column as your analytical column. Here are the hypothetical chromatogram acquired using C8 or C18 column. Assume that the peak widths are all 0.5 minutes. T0 is the retention time of solvent peak, and TRA and TRB are the retention times of analytes A and B. For C8 column, the solvent peak arrives at 0.5 minutes, where TRA and TRB are 2 and 2.8 minutes respectively. According to the equations introduced in the previous slides, the analyte A's retention factor is 3, and the resolution value between the analytes A and B is 1.6, which is close to the baseline resolution. For C18 column, the solvent peak arrives at 0.8 minutes, where TRA and TRB are 2.4 and 5 minutes respectively. Again, we can calculate the analyte A's retention factor and the resolution value of 5.2. If you'd like to achieve a better separation between analytes A and B, then you may choose the C18 column as your analytical column. If you'd like to have a greater retention factor of analyte A in order to reduce the potential matrix effect, then you may select C8 column as your analytical column. Using retention factor and resolution value will help you choose the appropriate analytical column. Next, we can further optimize the ionization parameters. Take electrospray ionization LC mass as an example. we can optimize the spray voltage, gas pressure, gas temperature, and the capillary temperature. Spray voltage would pose a huge impact on the overall ionization efficiency. If the applied voltage was too high, then corona discharge could occur and thus the peak signal would be drastically reduced. Corona discharge is an electrical discharge brought down by the ionization of a fluid surrounding a conductor, which occurs when the potential gradient exceeds a certain value. In the optimal condition, the ionization should be in the axial spray mode, which will result in the best peak signal. Rim emission is a phenomenon where small droplets at the rim of the spray cone emit ions that are different from the main spray. This occurs due to the presence of charged droplets that are surrounded by a thin layer of solvent, creating a double layer of charge. The double layer of charge creates a potential difference, leading to the emission of ions from the rim of the spray cone. This phenomenon can have a significant impact on the mass spectrometry analysis of biological samples, as it can lead to the formation of non-covalent ion clusters, which can interfere with the identification of the target molecule, causing drop in peak signals. Each mass instrument would have its recommended initial settings of ionization parameter. On the left, this table provides a recommended initial heated ESI setting in the thermal LTQ mass spectrometer. On the right is the typical ion source parameters provided by Agilent. These initial parameters are the empirical values to start with, but not the optimal values. We can manually acquire the optimal ionization parameter with an appropriate experimental design. Different LC flow rate and solvent proportions will affect the ionization efficiency. Therefore, it is better to optimize the ionization parameter after finalizing the LC gradient. This slide provides an example of acquiring the optimal ionization parameters by comparing the peak area values. In this method, we simultaneously detected the tetrachloroethylene metabolites from the glutathione conjugation pathway, glutathione conjugate TCVG, cysteine conjugate TCVC, and N-acetyl cysteine conjugate NAC-TCVC. By testing different values of a parameter in triplicates, we supposed to have a bell-shaped distribution in the peak area values where the optimal parameters were acquired. As these metabolites were detected in different time segments, we were able to assign the optimal parameters for each metabolite. Using this approach, we can find the optimal ionization condition for the best sensitivities of all analytes. Lastly, we can also optimize the mass parameter, or specifically the SRM, NRN transition parameters, including precursor product ion pair and their optimal collision energy. Usually, vendors will provide their commercial software that can get the optimal mass parameters for the analytes. Essentially, this software automated the workflow we introduced in the previous slides and report the optimal parameters with the greatest peak intensity. We have finalized the experimental condition of our method. How do we evaluate the performance of an analytical method? There are two analytical method validation guidelines for your reference, one from the US FDA and the other one from the European Medicines Agency. In this lecture, we will introduce the critical elements of method validation based on the US FDA's Bioanalytical Method Validation Guideline. The purpose of bioanalytical method development is to define the design, operating conditions, limitations, and suitability of the methods for its intended purpose, and to ensure that the method is optimized for validation. Before the development of a bioanalytical method, you should understand the analyte of interest, such as determine the physical chemical properties of the drug, in vitro and in vivo metabolism, and protein binding, and consider aspects of any prior analytical methods that may be applicable. Method development involves optimizing the procedures and conditions involved with extracting and detecting the analyte. Method development includes the optimization of the bioanalytical parameters listed in these slides to ensure that the method is suitable for validation. In the following slides, we will introduce each component in details. First is the reference standards and critical reagents. You should appropriately characterize and document, for example, determine the identity, purity, and stability of all reference standards and critical reagents, and store them under defined conditions. The purity of reference standards used to prepare calibrators and QC samples can affect the study data. Therefore, you should use authenticated analytical reference standards with known identity and purities to prepare solutions of known concentrations. The reference standard should be identical to the analyte. However, when this scenario is not possible, you can use an established chemical form, such as a free base, free acid, or salt of known purity. You should provide the certificate of analysis, including the source, the lot number, and expiration date for commercially available reference standards. For internally or externally generated reference standards that do not have a COA, you should provide evidence of the standard's identity and purity in addition to the source and the lot number. When using expired reference standard, you should provide an update COA or re-establish the identity and purity of the standard. If the reference standard expires, you should not make start solution with this lack of standard unless the standard's purity is re-established. For internal standards, you does not have to provide a COA or evidence of purity if it demonstrates that the IS is suitable for the specific use, like a lack of interference with the analyte. During the method development, you should choose the quantitation range of the assay and the concentrations of the calibration standards on the basis of the concentration range expected in a particular study. You should use the simplest model that adequately describes the concentration response relationship, as well as an appropriate weighting scheme and regression equation. When the method is validated, the calibration curve should be continuous and reproducible. You should prepare the calibration standards in the same biological matrix as the samples in the intended study. Study samples may contain more than one analyte. You should generate a calibration curve for each analyte in the sample. When surrogate matrices are necessary, for example, the artificial urine, you should justify and validate the calibration curves. The requirements for the calibration curves, including the lower limit of quantification, upper limit of quantification, as well as the acceptance criteria, are summarized in this table. Quality controls are used to assess the precision and accuracy of an assay and the stability of the samples. This figure illustrates the meaning of accuracy and precision. High accuracy means that the measurements are on the target, and high precision means that the measurements are close to each other. The ideal analytical method should be both accurate and precise. You should prepare the QC sample in the same matrix as the study samples to be assayed with a validated method. Freshly prepared QCs are recommended for precision and accuracy analysis during the method development, as stability data are generally not available at this time. During method validation, QCs evaluates the performance of a method and the stability of an analyte. Performance QCs are included in validation rounds to determine the precision and accuracy of the method. Stability QCs evaluates the stability of an analyte under various stress conditions, which will be introduced in the later slides. You should prepare any calibration standards and QCs from separate stock solutions. However, if you can demonstrate the precision and accuracy in one validation run using calibrators and QCs prepared from separate stock solutions, then you can use calibrators and QCs prepared from the same stock solution in subsequent runs. You should make up calibrators and QCs in lots of blank matrices that is free of interference or matrix effects. Evaluating the accuracy and precision across the quantitation range during the method development is essential to determine whether the method is ready for validation and involves analyzing replicate QCs at multiple concentrations across the assay range. Accuracy is defined as the determined value over true value, and precision is defined as the ratio of standard deviation over mean. Specifically, you should evaluate the performance at the LLOQ, low, mid, and high QC samples to determine if the method is suitable to analyze study samples. LC mass method validation experiments should include at least 3 independent runs and 5 replicates per QC level. The yellow shades highlights the passing criteria of a QC sample's accuracy and precision value, where it is 15% for the regular QC concentration and 20% at LLOQ. During method validation, you should verify that the substance being measured is the intended analyte to minimize or avoid interference. Selectivity of the method is routinely demonstrated by analyzing blank samples of the appropriate biological matrix, for example plasma, from multiple sources. For LC mass assays, matrix effects such as ion suppression, ion enhancement, or extraction efficiency should be evaluated. Internal standards should be assessed to avoid interference with the analyte. Potential interfering substances in the biological matrix, including the endogenous matrix components such as metabolites, decomposition products, and from the actual study, concomitant medications and other xenobiotics. If a stabilizers or enzyme inhibitors is used during sample collection, you should evaluate the potential for interference on the quantitation of the analyte. During validation, you should confirm that the assay is free of potential interference substances, including the endogenous matrix components, metabolites, anticipated concomitant medications, etc. Typically, you should analyze blank samples of the appropriate biological matrix from at least 6 different sources. Carryover between samples can occur in analytical methods. You should eliminate any carryover effects during method development. If carryover cannot be eliminated, you should assess the impact of any carryover during method validation on the accuracy of the study sample concentrations. The LLOQ defines the method sensitivity and should be determined during method development. The method should be developed and validated such that it will be able to meet the requirements necessary for the intended study samples. The LLOQ evaluations can be done separately or as part of the precision and accuracy assessment for the calibration range. The specific recommendations to validate sensitivity are listed in this table. Recovery refers to the extraction efficiency of an analytical process, reported as a percentage of the known amount of an analyte carried through the sample extraction and processing steps of the method. Recovery need not to be 100%, but the extent of the recovery of an analyte and of the internal standards should be consistent and reproducible. You should perform recovery experiments by comparing the analytical results of extracted samples with corresponding extracts of blanks spiked with the analyte post-extraction. During method development, you should determine the chemical stability of the sample. You should assess autosampler, benchtop, processed or extract samples, freeze-thaw, stock solution, and long-term stability of the analyte. For these experiments, you should evaluate the stability in the same matrix as that intended for in-study samples. However, when the matrix is rare, you can explore the use of suitable surrogate matrices such as artificial urine. Autosampler stability is the stability of the analyte in the process sample under the conditions in the autosampler, usually at 4 Celsius degrees. You should demonstrate the stability of extracts in the autosampler only if the autosampler storage conditions are different or not covered by extract stability. The benchtop stability is the stability of an analyte in the matrix under conditions of sample handling during the sample processing. You should determine the stability of samples under the laboratory handling conditions that are expected for the study samples. For example, the stability of samples maintained at room temperature or stored in an ice bucket. Extract stability assess the degradation of the processed sample relative to the starting material. You should assess the stability of processed samples, including the residence time in the autosampler against freshly prepared calibrators. Freeze-thaw stability refers to the stability of the analyte in the matrix upon freezing and thawing. You should assess the stability of the sample after a minimum of 3 freeze-thaw cycles. QC samples should be thawed and analyzed according to the same procedures as the study samples. QC samples should be frozen for at least 12 hours between cycles. Freeze-thaw stability QC should be compared to freshly prepared calibration curves and QCs. Long-term stability assesses the degradation of an analyte in the matrix relative to the starting material after periods of frozen storage. You should determine the long-term stability of the sample over a period of time equal to or exceeding the time between the date of first sample collection and the date of last sample analysis. The storage temperature studies should be the same as those used to store study samples. Long-term stability QCs should be compared to freshly prepared calibration curves and QCs. A stock solution refers to an analyte in a solvent or mixtures of solvents at a known concentration, which is used to prepare calibrators or QC samples. Stock solutions should not be made from reference materials that are about to expire unless the purity of the analyte in the stock solution is re-established. When the stock solution exists in a different state, for example, a solution versus a solid, or in a different buffer composition from the certified reference standards, you should generate stability data on stock solution to justify the duration of stock solution storage stability. If the method measures diluted samples, the integrity of the dilution should be monitored during the validation by diluting QC samples above the LLOQ with matrix to bring to within the quantitation range. The accuracy and precision of the diluted QCs should be demonstrated where the values within the plus or minus 15% are acceptable. Dilutions used during the validation should mimic the expected dilutions in the study, 5 replicates per dilution factor. Calibration curves and QC should be included in all analytical rounds. The QC should cover the expected study samples concentration range. Typically, the same curve fitting, weighting, and goodness of fit determined during the validation should be used for the calibration curve within the study. Change in the response function relationship between the validation and study sample analysis indicate potential problems. Total numbers of QC should be at least 5% of the total samples analyzed, or at least 6 samples, whichever is greater. If the study sample concentration are clusters in a narrow range of the standard curve, additional QC samples should be added to cover the sample range. If the additional QC concentration are not bracketed by QC validated before the study, the accuracy and precision of the additional QCs should be demonstrated before continuing with the analysis. If the partial validation is acceptable, samples that have already been analyzed do not require reanalysis. The QCs should be interspersed with study samples during processing and analysis. In each analytical run, the lack of analyte interference at the LLOQ should be confirmed. The analytical run fails if the calibration or QC acceptance criteria are not met. If the bioanalytical method necessitates separations of the overall analytical run into distinct processing batches, for example, groups of samples processed at distinctly different times or by different analysts, each distinct batch should process duplicate QCs at all levels, along with the study samples. For example, when the number of samples exceeds the capacity of a 96-well plate, or when the solid phase extraction manifold cannot accommodate all samples. If there are dilution steps, study sample dilutions should use the same matrix. Assays of all study samples of an analyte in a biological matrix should be completed within the time periods for which stability data are available. The internal standard response should be monitored for variability, retention time shift, and its impact on the accuracy of the estimated unknown sample concentration, if any, should be addressed. All study samples from the subject should be analyzed in a single run, especially for study design with repeated measures from individual subjects. Incurred sample reanalysis (ISR) should be performed, where the recommendation and acceptance criteria are summarized in this table. Aside from the full validation, there are other types of method validation, including the partial validation and cross-validation. The partial validation evaluates modifications of already validated bioanalytical methods. Partial validation can range from as little as 1 intra-assay accuracy and precision determination to a nearly full validation. Raw data on partial validation should be retained at the analytical site for inspections when requested. Typical bioanalytical method modification or change that falls into this category include, but are not limited to, the following examples. Cross-validation is a comparison of validation parameters of two or more bioanalytical methods or techniques that are used to generate data within the same study or across different studies. Also, cross-validation is necessary when sample analysis within a single study are conducted at more than one site or more than one laboratory. In such cases, cross-validation with shared matrix QCs and non-pooled subject samples should be conducted at each site or laboratory to establish inter-laboratory reliability. Pooled incurred samples can be used when insufficient volume exists. An SOP or validation plan should define the criteria a priori. Okay, that's for today. In the next lectures, we will talk about the non-targeted analysis, NTA.